तो चलिए समझते हैं. Welcome everyone to today's study breakdown. Right now, we're diving deep into the absolute core of biochemistry. We're looking at Chapter 4, which is all about proteins and how we actually determine their primary structure. Trust me, अगर आपके concepts यहाँ पर clear हो गए, then everything else in molecular biology is going to become so much easier. So let's just jump right in.
और देखो यार, we aren't just pulling this information out of thin air. We're breaking down the exact text from the 32nd edition of Harper's Illustrated Biochemistry. As you probably know, this book is literally the gold standard for medical students globally. हम इस टेक्स्ट की एक-एक important line को decode करेंगे ताकि आपकी exam prep बिल्कुल effortless हो जाए. Just think of me as your senior, kind of guiding you through this whole journey.
Okay, फटाफट अपना agenda देख लेते हैं. First, we'll cover the basics of protein structure. Second, separating proteins with chromatography. Third, cleaving the disulfide bonds. Fourth, checking purity using SDS page. And finally, why this all actually matters medically.
All right, Section 1, Basics of Protein Structure, the 4 orders.
So, सबसे पहले जब हम proteins को study करते हैं, what exactly are we looking at? Like, how is this massive molecule actually organized? Well, Harper's defines this using what's called the 4 orders of protein structure. ये basically a conceptual framework है. It describes the complete molecular nature of how a protein is synthesized, how it folds, and how it ultimately assembles into a functional unit. ये concept समझना बहुत ज़रूरी है guys, because protein architecture is highly ordered. It is definitely not just some random jumble of molecules.
Let's break these 4 orders down exactly as they appear in the book. First up is the primary structure. This is simply the sequence of amino acids in the polypeptide chain. Just the sequence. Then comes secondary. This is the folding of short, contiguous segments. We're talking specifically 3 to 30 residues here, into geometrically ordered units. Tertiary is the assembly of those secondary units into larger functional domains. And finally, quaternary. This is the arrangement of multiple subunits. So, for example, a protein might be a homotetramer, like alpha-4, or it might have a mix of subunits, like alpha-2, beta-2, gamma. Basically, sequence select or assembly to every single step is absolutely crucial.
Now, obviously, visualizing all those thousands of atoms in a 3D protein is way too complex, so textbooks use schematic diagrams. ध्यान दीजिए यहाँ पर, pay close attention to how we simplify these structures. We use ribbon diagrams to basically trace the polypeptide backbone. When you see spirals, those indicate regions of alpha helices, and when you see arrows, well, those indicate regions of beta sheets. ये visual shortcuts हैं, और ये आपको 3D structure तुरंत समझने में बहुत मदद करता है.
Moving right along to Section 2, separating proteins with chromatography, isolating the target.
Right, so now that we have our structural concepts locked in, अगर हम एक specific protein को isolate करना है, we first have to separate it out from a really complex biologic sample. And this is exactly where liquid chromatography comes into play. Imagine this is a flowchart in your mind. Step 1, reservoirs hold your mobile phase liquids. Step 2, microprocessor-controlled pumps and a mixing chamber control the fluid flow. Step 3, you load your sample, which is the analyte, right into the injection port. Step 4, the liquid travels through the column, which houses the stationary phase matrix. This is literally where the actual separation magic happens. Step 5, a detector analyzes the output. And finally, Step 6, the fraction collector gathers the eluent liquid in separate little tubes. Memorize this flowchart, guys. It's practically the heartbeat of protein isolation.
Okay, Section 3. Cleaving the disulfide bonds, breaking down the chains.
Fantastic! We've isolated our target protein. But wait, proteins are often folded up tightly and held together by these really strong covalent bonds. Chains ko todna parega. We absolutely need to break apart those adjacent polypeptide chains so we can sequence them properly. And hey, this brings us to a classic exam question. How do we cleave disulfide bonds? Well, there are 2 main ways, and you absolutely must know the difference. On the left, we have oxidative cleavage, which uses performic acid. This specific reaction yields cysteic acid residues. Now on the right, we have reductive cleavage using beta-mercaptoethanol, and this one yields cystenyl residues. Ye exam ke liye bahut important hai yaar. Oxidative gives you cysteic acid, while reductive gives you cystenyl. Lock these chemical outcomes into your memory right now.
Section 4. Checking purity using SDS page, analyzing the mass.
So, moving on to our next critical lab technique. We isolated the protein, we cleaved it, but hum check kaise karenge ki sample actually pure hai? Like, how do we verify it? Well, we use a brilliant technique called SDS page. This method is amazing because it lets us visually observe the successive purification of our proteins. After we run the gel, we stain it with a dye called Kumasi blue so we can actually see the protein bands. And here is the really crucial part. We rely on standard markers. These markers act exactly like a ruler, allowing us to compare and measure our purified protein's mass in kilodaltons, or kDa. Samjhe? It's basically a molecular weighing scale.
And finally, Section 5, why this matters medically, the clinical application.
Okay, so we've talked a lot about structures, running chromatography, cleaving bonds, and staining gels, but let's just take a step back for a second. As future doctors and scientists, you really need to know the why. Ye sab lab work human health ke liye zaruri kyun hai? I actually want to read you this direct quote from Harpers because it just perfectly bridges the gap between the biochemistry lab and real-world clinical diagnostics. An important goal of molecular medicine is to identify biomarkers, such as proteins and/or modifications to proteins whose presence, absence, or deficiency is associated with specific physiologic states or diseases. That is the ultimate goal, guys. We do all of this tedious lab work so we can find that exact protein marker that tells us if a patient has a disease, often long before the actual symptoms even show up.
And that brilliant reality brings us to the end of our session today. But before you go, I want to leave you with a slightly provocative question. We've mastered the classic textbook methods today, but soch ke dekho? How might modern genomics, advanced computer algorithms, and massive mass spectrometry databases revolutionize our search for these life-saving biomarkers in the future? Technology is evolving so rapidly right now. Keep thinking beyond the textbook, keep pushing those boundaries, and I will see you in the next breakdown.